This is a summary of one of the most recent projects we did with one of my favorite collaborators, who also happens to be my dad, Georgiy Karev. And the title of the project is Energy Constraints and Neural Strategy Transitions in Alzheimer's, a game-theoretic model. So let's start with some background. The term Alzheimer's disease was originally referring really to only early onset cases. That was described in 1907 report by Alzheimer. Over time, those shared features like amyloid plaques and tau tangles were observed in both younger and older patients with dementia. And this led to merging of early and late onset cases under the single diagnosis of Alzheimer's disease. But actually, this may be conflating two distinct conditions. Early onset Alzheimer's, which is largely genetic, it's linked to some specific mutations that were identified in the 90s. And it is highly penetrant, which means that if an individual happens to have these mutations, they're very likely to develop this disease. But it also accounts for less than 5% of all cases. Late onset Alzheimer's used to be known as senile dementia. Now we're just calling it late onset Alzheimer's. That's the one that makes up the majority of the cases, up to 95% of all the cases. It is not caused by a specific mutation. The ApoE4 allele does seem to be associated with higher risk. It seems to be connected with higher inflammation. And if you want to get some excruciating detail on it, I put together a presentation that really goes into all the background that I've collected in research for this project. I will put a link to it, and you can also easily find it. It's not fully penetrant. Even carriers of 2 copies of ApoE4 allele are not guaranteed by any stretch of imagination to develop the disease, but they are more likely to. And it is understood to be influenced by, yes, some genetic, but really environmental and lifestyle factors, and that's the one that I'm going to be talking about. There exist multiple hypotheses about the origin of Alzheimer's and the initiating events. And the one that really resonates with me and that makes the most sense to me is the metabolic and mitochondrial dysfunction hypothesis. The idea that Alzheimer's is actually a metabolic disorder in the brain. Again, I go through all of the information that I was able to find and understand in this other larger biological paper. So here I'm just going to give a snapshot of it, and you're welcome to go and look in more detail in the other work. The idea here is that Alzheimer's may arise from metabolic and mitochondrial dysfunction in the brain. This idea was championed, to my knowledge, primarily by researchers like Jack de la Torre, but it is supported by metabolic imaging studies. I'm going to show an example shortly. And the idea of the underlying mechanism is that there is a mismatch between brain energy demand and supply. So you might have impaired glucose and oxygen delivery or utilization, which may lead to disruption of neurovascular coupling and mitochondrial function. And ongoing energy deficits can initiate a pathological cascade, which will then result in increased tau hyperphosphorylation, amyloid accumulation, synaptic failures, all of those things that we associate with dementia, and particularly with Alzheimer's disease. But the idea here is that these are maybe consequences of metabolic failure rather than initiating events. And then if you're taking it further, then maybe types of deficits will depend on where in the brain this hypometabolism, so this mismatch between energy demand and supply, will occur. And maybe different patterns will yield different dementias, like depending on where in the brain you might have deficits. That's where you will start seeing the particular issues of Alzheimer's versus frontotemporal dementias versus others. So this was a very, very exciting and striking experiment that was done by Mosconi et al., or published by Mosconi and colleagues in 2008. And in it, the authors used PET imaging to assess glucose metabolism in the brain. So basically, they're injecting a radio-labeled glucose, and they are then tracing which parts of the brain light up. And the areas of high glucose uptake will be brighter, and areas of low glucose uptake will be dimmer. And the prediction is that in the brains of Alzheimer's patients, you would have more hypometabolism, so insufficient glucose metabolism. Okay, so this is a figure from this paper. They normalized, so the Z-score, they normalized the visualization to highlight the areas of hypometabolism. So these are the areas where you do not actually get enough glucose absorption. kind of reverse of what I just said, but the visual here is normalized in order to see the areas where you are not getting enough glucose metabolism. And you do not need to be a neuroscientist to really see the patterns here. So AD is Alzheimer's, MCI is mild cognitive impairment. It's kind of the pre-Alzheimer's. So these are the individuals who have transitioned to Alzheimer's disease. And on the bottom are the individuals who did not, so who stayed relatively stable. And you can see, of course, clearly that individuals with Alzheimer's have severe hypometabolism, so a lot of regions of the brain where these parts of the brain are not getting enough glucose. These individuals, on the contrary, have maybe very small regions of the brain where this is happening. And this intermediate state of individuals who kind of started here but then progressed to full-blown Alzheimer's, of course, you can see that, okay, they're exactly in between. So one of the interesting implications of this is also that maybe these changes precede clinical diagnosis, so supports the idea that these might be the initiating events, that mismatch between supply and demand of nutrients in the brain when taken, and which happens long enough, then actually then results in development of the Alzheimer's phenotype. And this is the paper if you want to look it up. Okay, so that was part one of the background. And now we're going to part two about strategy selection and group size. And really the question here is that, well, if you have hypometabolism, if you have issues with nutrient supply, then how would neurons adapt their communication strategies under metabolic stress? Because here you're really dealing with this fundamental trade-off of conserving energy or using the energy that you have and transmitting high fidelity information. Neurofiling is very metabolically expensive, of course, especially if you want to maintain high signal-to-noise ratios. So you can really imagine this trade-off of using the resources to maintain high quality of communication or conserving the resources but then losing the high quality of communication. The really interesting piece here that I found and that really kind of tied this thing together was about group size. And there was this really interesting paper that was showing that larger neuron groups are more likely to have better signal fidelity, but also higher energy costs. I mean, again, it makes intuitive sense, but it is helpful to see it and to formalize it. And smaller groups maybe will be more energy efficient, but they would have less information, reduced information quality. You will have less opportunity for redundancies, frankly, but those redundancies might actually compensate for and ensure higher signal-to-noise ratios. And so the prediction based on this would be that if you have ample energy, then maybe selection will favor larger, more interconnected groups of neurons with high information fidelity. And if you are in a situation of metabolic stress, then maybe selection may shift towards smaller energy conserving groups with low information quality. Okay, so this is our prediction. And now let us discuss how we're actually going to formulate it. And we're going to use a game-theoretic framework where we're going to look at neurons as players in a game. And each player can choose between two strategies during interactions. You can be energy efficient, or you can have really high quality information transmission. And so if you were to put it in a matrix, then you would have, yes, your neuron A versus neuron B, the classic payoff matrix, and these would be the coefficients in it for their interactions. And the payoffs in this classic payoff matrix, we are proposing that they may represent the so-called utility function, which would be a balance of information gain versus metabolic cost for each interaction. And we are going to define utility function as some weight 1 of information gain minus some weight 2 of metabolic cost. And so we are going to then ask that, so first we're going to look at this. So, okay. Then this would be how we would interpret these coefficients, what they gain versus what they lose and what it costs them. And the question then, because this would be just a classic game theory analysis, but we want to know what will happen if the coefficients are not fixed, but could be affected by external factors, such as glucose. But we're going to get to glucose first. We're going to go through a different example. And the question would be that we're going to look at, could this, so modifications to these payoff coefficients, affect group sizes, so the groups in which neurons work together, with the implication that larger groups are less metabolically efficient, but they have higher information quality. Smaller groups would have maybe smaller energy demands, but also poorer quality of information. Okay. So in order to look at that, we are going to introduce the so-called switcher function. And the hypothesis is that if we change some of these payoff coefficients, then the system will evolve towards smaller groups, or towards different side groups, larger groups or smaller groups. So we are going to introduce this kind of a switcher function. It has a very particular property that it is basically going to be positive before switch time t and is going to be negative after switch time t. And t would be just after switch time, when t is larger than the switch time. And the idea then would be that we had this kind of, so this was our previous payoff matrix, and we are going to replace it, and then we're for this particular illustration, we're going to put this switcher function on these coefficients, but you can put them anywhere. Okay, what does it actually mean? Well, let's assume that we have a case. This is an example case when the switch time, so this switcher function occurs at when time is equal to 100, whatever units we have. So what we have is that this switcher function is going to be positive. Positive tau will affect the steepness, of course, of this curve. And then around here, okay, it starts to go down, and then around 0 it changes signs, and now we're going to get 0. Okay, so that would mean that over time, so this would be like a control. Yes, so if there is no switcher function, then at time 0, we have the population just keeps evolving as it is evolving. In this particular formulation, parameter beta refers to is a proxy for group size. This is all written up. I'm not going to go into mathematical detail because I believe that might get confusing. I just really would like to get the main point across here. But it is all written up, and there will be a reference in the end. But the idea is that, just take my word for it right now, when beta is 0, then we have larger groups, the way that the beta is defined, 1 over group size, population size. So is it going to 0? That means group size is increasing. And then this is limited to 0.5. So when beta is 0.5, that means smaller groups. Again, just take my word for it, and please look at the derivation in the paper. Okay, so now we are actually adding the switcher function here. I am now turning the switcher on. And this is showing exactly what I was hoping it would show, which is that at time equals 0, then we have a switch. So what happens is, as this value goes down, so up to over here, these two groups, they behave the same, but then this is starting to go down, and so our distribution of different strategies of group sizes in the population is going down. And once the switch has occurred, now instead of having the majority of the groups being large with high information fidelity, but also high metabolic costs, we have a bifurcation. We are going through to a stage when most of the neural groups are actually becoming smaller, so they become low-cost energy saving. That's the interpretation here. Okay, so and we played with different with placing the switcher on different coefficients, and we played with different values of the coefficient. Again, everything is in the paper. This particular exercise is in the appendix, but I encourage you to take a look if this is something that's of interest to you. Now, the question is, what could influence this switcher? Because, again, we're going through this whole neural group sizes, and hopefully it's becoming very clear where we're going in terms of, maybe larger group sizes of neurons are better connected, and maybe in the areas of hypometabolism, now you have energetic constraints. You might start selecting for smaller groups, and because they do not have enough nutrients, that will have poorer quality information, and maybe that can be interpreted as initiation of cognitive decline. Not a very complex thought, but this is kind of what we're looking at. So what could influence a switcher? What could really cause this dramatic transition? Well, one of the topics that has been of great interest to me recently is metabolism and type 2 diabetes. And there is going to be another recording of work on emergence of type 2 diabetes that is going to be also linked up here. But the main background information is that type 2 diabetes correlated with increased risk of developing Alzheimer's disease. Even more than 10 years ago, for example, there was this very nice meta-analysis, and they were looking at an association between diabetes and risk of either old-type dementia or Alzheimer's vascular dementia, which within the framework that we're discussing would really be a spectrum of maybe same-ish set of conditions, but you would have metabolism deficits in different parts of the brain. And the results of this analysis showed that 73% increased risk of all-type dementia, 56% increase in risk of Alzheimer's, 127% risk in vascular dementia in diabetes patients. So this could be a very good place to start looking. And even if you look through the literature, Alzheimer's disease has even been referred to as type 3 diabetes. But how would that actually work? I mean, if you have high blood sugar systemically in the body, why would that translate into low sugar and diminished metabolism in the brain? Excellent question, gentle reader. So here is one of the processes where how this could be taking place. I'm not going to be modeling all of these, but I wanted to go through how exactly this high blood sugar systemically can translate to low actually functional glucose in the brain. So you can have high blood sugar or chronic oscillating hyperglycemia, so high blood sugar. It can do a lot of things. But one of the things that it can do is it can cause dysregulation of glucose delivery systems. Like, for example, the blood-brain barrier is lined with insulin-independent glucose transporters like GLUT1, and they are adaptable. So if there is a lot of glucose, then maybe they will have lower expression. If there is low glucose, then they will express a little bit more. The idea is that this is a system to really maintain glucose homeostasis in the brain. And so it will adapt, but it is not designed to sustain really big oscillations. And when this keeps happening, this transporter expression regulation might have issues. In addition, so when sugar binds to, there's a lot of floating sugar binding to proteins and sugars, it can create these complexes that can compromise the structural integrity of the blood-brain barrier, so causing it to become leaky. And the leaky blood-brain barrier with dysregulated glucose in the brain can be a big problem, not only because it cannot now regulate the glucose concentrations in the brain very well, but also can let in a lot of bad things like inflammatory markers, like toxins, and a lot of things like that. Also, now we have high blood sugar. We know that in the rest of the body just as well, of course. It can cause vascular issues. Well, it does the same thing in the brain. You might get damage to small blood vessels in the brain, so that might compromise glucose delivery. The transport lines are now compromised. You can have some extraneural inflammation, multiple pathways for it. Could be things that are getting in from the body through the now leaky blood-brain barrier. Could be some other infections that are getting through, which are causing inflammation. Could be things that mitochondria are not responding very well to oscillating between high glucose, low glucose. That can cause mitochondrial damage. When mitochondria are tucked away inside the cells very happily, the immune system does not recognize them as the bacterial ancestors that they were. Because, of course, long time ago, we entered in a symbiotic relationship with bacteria, which now became our mitochondria. But if you have a lot of mitochondrial damage and you have a lot of cells not supporting their life, mitochondria might start spilling out, and the immune system will start recognizing them as, this is bacterial in origin, I should attack that. That can also lead to creation of reactive oxygen species, and just a lot of feedbacks of that. That can in itself trigger amyloid accumulation, tau tangling, and a lot of things. If you want excruciating detail on these and more, so feel free to see this write-up as I went down a rabbit hole on this topic. And as I said, there's also about an hour-long presentation on this Many Roads to Dementia paper. But all of this can lead to inability of neural cells to generate sufficient ATP to function. That's really the point that I wanted to make here, as a connection between how systemic high blood sugar can trigger issues with functional glucose in the brain. Okay, so now I would like to introduce the idea of this glucose-dependent switcher. The idea, that is, before we were just having a basic time-dependent switcher for illustration that, oh, this occurs as a function of time. When the time surpasses some switch time, then tada, this switches the value of the coefficient. The adaptation and the modification that we're looking at here, what if you take the same function, so same idea, but this is glucose dependent. That is, you have some dynamic glucose. Here we're kind of going to be estimating fasting glucose, and we have some kind of a threshold. And the switch will occur when glucose levels, so systemic glucose levels, let's assume fasting glucose, so long-term, exceed this threshold. And the threshold could be individual for different people. And so if we're going to look at our updated payoff matrix, it would look something like this. If you remember, this was before A times plus, for example, f of t, and now it's in the glucose switcher. So this guy over here. Okay, so what would that actually look like? So just to have a little bit of a background, normal fasting glucose ranges, because I'm trying to parameterize this to actually be somewhat in the physiologically reasonable range. This is not obviously a detailed mechanistic model, but I'm trying to make sure that it is at least within reasonable ranges. So normal fasting glucose, about 3.9 to 5.5 millimole per liter, millimolar, then pre-diabetes between, say, 5.6 to 6.9, diabetes is over 7. And just to recall, within the frameworks here, small beta, so beta going to 0, translates, the meaning is it's larger groups, which we interpret as having higher information fidelity. Beta going to 0.5 is smaller groups. They could be energy saving, but also lower information fidelity. Okay, let's look at actually a couple of examples. So assume that I have an individual whose individual threshold, this G threshold value, is 7.5 millimolar, so deep in the diabetes, or what is considered diabetes territory. So this could be something that we would end up with. So this is my switcher function. As you can see, yes, it is starting to decrease here, but it never actually, and I'm looking at like 100 years, but it never reaches 0. So the sign never flips. It doesn't technically have to be 0. It's just something that affects the values of the coefficients. It just here we parameterized it in such a way as to make this very visual, that it's very clear when the switch occurs. So we made it to be when this happens at 0, but it doesn't have to be. This is kind of one of those modifications can be taken forward in the future. And then we have a coupled system of equations for increasing glucose over time, so emergence of type 2 diabetes. And over time, maybe you have this individual, their glucose is increasing, but it never reaches this threshold level, so the switch never occurs. So this person probably is going to be fine. Okay, now let's assume we have another person whose individual threshold is at 7.15 millimolar. Okay, so now we have something interesting going on here. So at about 75, 77, whatever this value is, this is when my switcher function crosses 0. It crosses 0 also, you can see at the same time, that's when my baseline glucose crosses this threshold of 7.15. And now let's look at this guy. Again, beta equals 0. This is kind of what a healthy person would be, that all of their neural groups remain large and love information fidelity. What you start seeing here is, okay, yes, but as this is going down and down, this is starting going down. Yes, the majority of the groups are still large and interconnected, but once we hit this bifurcation point, essentially, once we hit this switcher threshold, then most of the groups are now becoming smaller. So the population of neurons, basically the strategy of balancing out the utilities, the information, the need for high information fidelity versus metabolic costs, essentially, it's no longer worth it to increase in high fidelity. And so most of the groups actually become smaller. Ouch. So here, this switch occurs, the simulated switch occurs around 75 years old. If I lower it even more, again, assume somebody who is super sensitive to it. Well, this now happens even sooner and sooner. I mean, you can see the point. You can predict it from here. So yes, so this decline is starting to occur maybe a little bit sooner. In this particular case, the switch occurs around 60 years old, as does over here. And this is when you go from having larger, primarily larger, very nicely connected groups, then to most of them becoming smaller and less connected because the metabolic stress is just too high to maintain large interconnectedness. Okay, so what are some of the implications of the model? And this is kind of the idea that really we're just trying to think through this here, that maybe cognitive decline within this framework may reflect a strategic shift. So the neural cells are changing their strategy of how they're going to connect with each other. And under metabolic constraints, neurons may be adaptively downshift to smaller, more energy efficient networks, but they are not the ones that actually have high information fidelity. And maybe one can think of this as group size evolution, linking this more systemic metabolic dysfunction to local neural behavior, with energy abundance being connected and leading to or supporting large, robust, high-fidelity networks. Yes, they are costly, but there are resources for them. But if you end up in areas of increasing hypometabolism, as we do see in for example, in the PET images, and eventually, maybe you get to a breaking point, and the strategic shift happens where now energy scarcity leads to smaller, minimal cost, yes, but also low fidelity networks. Also, there are some, Alzheimer's doesn't, or just onset dementia doesn't just happen. There is a prodromal phase. There's a time that precedes the abrupt changes, and even if you know anybody, if you work with anyone, you know that the decline initially is slow, and you're not entirely sure if it's happening. Then it is happening, but it is still being compensated maybe by the other parts of the brain, and then it just shifts, and things go downhill very quickly. So maybe our model can propose also capture the idea of this prodromal phase, where you have gradual shifts in strategy before the visible decline, and might have this threshold effect, when you might end up having rapid collapse in network fidelity once the metabolic stress surpasses some tipping point. This was a paper from a couple of years ago, kind of summarizing also what I'm talking about here, that there is maybe a preclinical stage, and so maybe some of the things are starting to show up. Again, they are focusing on amyloid beta. My understanding of all of this right now is this is a consequence of metabolic decline. Again, please go look through the my giant treatise on dementia, just trying to put all of this together and make sense of it. And then, okay, here maybe you're starting to already, the hypometabolism becomes problematic enough, you're starting to develop cognitive symptoms until transition to dementia. And if maybe within the framework of what we have going on here, maybe you have this preclinical phase over here when you're just starting to go down, so the switch is starting to go down, and maybe you have this prodromal phase over here where again now this is really starting to accelerate, and then once you surpass the threshold, now you had a strategic shift going from large interconnected groups primarily, even though they're decreasing, to really small fidelity groups. Okay, so you can also expand this proposed framework. We are talking about how neurons shift strategies based on metabolic constraints and trade-offs. So this whole idea of energy cost and information fidelity. And in this particular example, we use glucose-dependent switchers, also on select PF coefficients. But it can be very adaptable. You can have other switchers that reflect inflammation and oxidative stress and neurotrauma and infection, all of these other things that are connected mechanistically to metabolic issues and to also the origins of a lot of dementias. Maybe you could reflect sensory deprivation. So maybe reduced impact of need for high information fidelity. When the cognitive function is not utilized enough. Maybe different people have different switcher thresholds, or they have them on different coefficients. We showed, again in this paper, that not all switcher placements lead to these changes, these dramatic transitions. We selected a case when we had set this on these two coefficients, but maybe they could be only over here, or maybe they could be over here. I'm not entirely sure where they would go, or maybe this is really truly individual. Maybe there's some various combinations that are individual specific. Maybe there are groups of individuals. This is something that could potentially be explored in more detail. Also, you can use this to maybe model pathogenesis and intervention. For example, right now we're putting the switcher on the entire utility function and we're not simulating interventions, but both the pathogenesis and intervention switchers could be placed on different coefficients or the different components of the utility function. If you remember, we have this idea of that there's this effect of information gain. So this is the benefit, I guess, that we get from high quality information, and these are the costs. And maybe you have some things in pathogenesis that would decrease this term. Yes, hyperglycemia is what we have. Could be maybe infections, could be vascular disease, could be some genetic predispositions. Or pathogenesis can be through increasing this term. And then maybe you might have, again, some of these things affecting that, the high metabolic costs, like, I don't know, immune cells out competing everyone for glucose because they're fighting infections all the time. And same idea with interventions. Maybe you have interventions to increase this term, like cognitive stimulation, like listening to music or dancing or solving puzzles or improving vision and hearing. Really increasing the value of this term of the utility function, or you can have interventions to maybe try to decrease this term, treating the infections, metabolic interventions, exercise. There are some other things like photobiomodulation therapy was an interesting thing that I was discovering. I don't know, clotho might do some protection on the insults. Just a lot of interesting things. Intranasal insulin is another thing that was really popping up. So there are multiple. The idea is that you can use this framework of the utility function to both simulate pathogenesis by affecting the weight of this term and the magnitude of either of these, and also the interventions, and trying to balance it out. And that is that. So here are some references. So this is the actual paper. I put it up as a preprint on arXiv, and hopefully soon it will come out elsewhere. This is my deep dive into just mechanisms of dementia that really got me interested. This is out as a preprint as well, and this has detailed background biology, history, different hypotheses about amyloid hypothesis, tau hypothesis, why I believe a metabolic hypothesis actually unites them all as an initiating event, etc. The mathematical details on group size selection. So this is a paper where a lot of the math has been developed. We put the key aspects of it into the main paper. But if you want more detail and more examples, this is of course the place to go. This is the model that I used for coupling it to this game-theoretic framework. So those increasing glucose functions, emergence of hyperinsulinemia and hyperglycemia, in this particular case, especially hyperglycemia. So this was the model that was used here, developed in this paper. Again, a summary is given in the appendix of the main manuscript for this project. And it also has a small modification of this energy constraints that is now given in our 2025 paper. And an important disclaimer, I'm not a medical doctor, I'm a PhD scientist, I'm not a neuroscientist. I primarily work in cancer. I got into it because I was interested in cancer metabolism and some interesting things there, and then just Yes, rabbit holes happen. Also, these are my personal views, do not necessarily represent views of my employer or any organization. This is personal research. No financial support was received, no financial interest to declare. This was just really interesting to think about and a very fun project to work on. Thank you very much for your attention.